Hi everyone, this is Kalyan Kumar and welcome back to Chemistry Universe. We've done three parts of proteins. We're doing biomolecules and this is part 4. And in this we're going to talk about peptides. And primarily we're going to talk about the chemical properties of peptides. Now peptides, if you remember from the earlier part that we did, a peptide bond is an amide linkage between the amino group of one amino acid and the carboxylic acid group of the other amino acid. So this amide linkage is the peptide bond. Now though when two amino acids combine, you get one peptide bond, we call it a dipeptide because basically there are two amino acids. So just imagine that whenever we say how many peptides, we'll simply look at the number of amino acids forming the bond rather than the number of bonds formed. Because for two amino acids, there's only one bond formed. We're not looking at the number of bonds, we're looking at the number of amino acids. So when I say dipeptide, 2 amino acids, tripeptide, 3 amino acids. So that is how we're going to address them. So let's look at some of the chemical properties of the peptides. Okay, part 4. So let's understand again what are peptides. Now you know that a peptide is an amide formed by intermolecular reaction of the amino group of one amino acid and the carboxyl group of a second amino acid. Now chemically, peptide linkage is an amide formed between COH group and NH2 group. So basically you have let's say glycine and you have another glycine, water is released and you get this. So this is the peptide linkage, the amide one. The product of the reaction is called a dipeptide because it is made up of two amino acids. For example, when carboxylic acid group of glycine combines with amino group of the alanine, we get a dipeptide. It's called glycylalanine. So this is glycylalanine. And it is usually denoted by this. So this is where the three letters come into existence to denote the polypeptides and the dipeptides, tripeptides and so on. If a third amino acid combines to a dipeptide, the product will be obviously a tripeptide. A tripeptide contains 3 amino acids linked by 2 peptide linkages. When the number of such amino acids is more than 10, then the product is called a polypeptide. And a polypeptide with more than about 100 amino acid having molecular mass generally higher than 10,000 is called a protein. However, the distinction between a polypeptide and a protein is not very sharp. So you can't really, ask a question whether this is a polypeptide or a protein because a polypeptide having a huge molecular mass is definitely a protein, but there is no particular demarcation for that. Now, some polypeptides with fewer amino acids also are likely to be called proteins. Now, these proteins ordinarily have a well-defined conformation of a protein such as insulin, which contains around 51 amino acids. Now, when two amino acids of similar type are used to make a dipeptide, then only one type of dipeptide is formed. That means glycine, glycine. There are only two possibilities here. When two different types of amino acids are used to form a dipeptide, 4 different dipeptides are possible. So you may wonder how 4. So if you look at A and B, amino acid A, amino acid B. So A can form a dipeptide with another A. B can form a dipeptide with another B. That makes it 2. A can form a dipeptide with B where the NH2 of A reacts and the COH of B reacts. And the other way would be COH of A reacts and NH2 of B reacts, and they're all going to be different. So that is how you get 4 possibilities of dipeptides. So if I have this, glycine and alanine, I can use this water up and I'll get something like this. So I can get a GLYLA. So I will always write that one first whose COH is used. On the left side, you will always write the one whose COH is used. Or you can write it like this. The abbreviated name of the dipeptide is written by first writing the 3-letter code of the amino acid with the free NH2 group. That means whose COH is used, followed by a hyphen, and then writing the 3-letter code of the amino acid with the free COH, that is which has used this NH2. The name of the dipeptide is written by writing the name of the amino acid with the free NH2 with the suffix "-ine" is replaced by "-ile", and then the name of the amino acid with free COH. So you can see glycylalanine. So if you can look at this name, we have called this glycylalanine. That means the one whose COH is used, or the one with the free NH2, that will end with YL. Instead of glycine, the INE, this INE goes and you get a YL. That is how you denote them. All right. So if we go on further, so the possibilities are glycine with glycine, and you'll get this, and you can call it GLY, or you can call it glycyl glycine. You can get alanine with glycine. You'll get this. You can call it ALA GLY or alanyl glycine, or you can get alanine and alanine, and you will get this, which could be alanyl alanine. So these are the others that you get. Now for a polypeptide, the same rule is applied from left to right with the free NH2 amino acid with the YL suffix, and eventually writing the name of the amino acid with the free COH group. Say for example, I have a tripeptide, 3 amino acids. As you can see, 2 of them are alanine, the first 2, and then you have the glycine. So you have ALA GLY for the short letter, or alanyl glycine. So all of them will get YL except the last one. Now if you want to create a specific dipeptide in which we use 2 different amino acids, and we want the NH2 of a specific amino acid to be linked to the COH of the other, and we don't want all the other possibilities, we can do this by blocking the groups that we don't want to be linked. So say for example, you have this, okay, and you have this. So I don't want the NH2 of this to react. So I'll first make it into an amide. Then I'll take the other one, and I don't want the COH of this to react. So I'll make an ester of this one. Then I will combine them, and I will get this. And then by hydrolysis, I'll be able to get back the NH2 here as well as the acid here. All right. Now, if there are M amino acids that need to form a polypeptide chain containing only N of those, then the total number of possible polypeptide chains are actually M to the power N. What this means is that if you have, let us say, 4 amino acids, okay, you have 4 amino acids, and at a time you want to combine only 2 of them. So how many possibilities can you get? And that is 4 to the power 2, which is 16. How? Because remember, the first spot, if there are 2 amino acids, the first spot can be taken by any of the 4. But the second spot can also be taken by any of the 4. So you will find as many 4s as there are number of amino acids used. So if the amino acid used are N, and the total amino acids in the beginning are M, then you'll have to write M N times for filling up all the spots. So these are the possibilities. So we're not saying this is the only one that is formed. So with that, we come to end of this video. That is the chemical properties of polypeptides. As you can see, this one was a short one, but very important one. So you need to understand how to get a polypeptide, what are polypeptides, and how do we denote them. So in the next one, we're going to talk about proteins. So I'll catch you then. And till then, goodbye. Have a nice day. And thank you for watching this.